Good morning. My name is Dr. Andreas Brunklaus. I'm a consultant pediatric neurologist at the Royal Hospital for Children in Glasgow. I'm very grateful for this invitation to talk to you this morning about SCN2A related disorders and what lessons can be learned from the sodium channel family. I have a long interest in research on sodium channel disorders, in particular SCN1A and Dravet syndrome. But I've also been looking at the sodium channel family, and I think that there are indeed some lessons that can be learned from other sodium channels that will also apply to SCN2A related disorders. So thank you for this invitation. And I would like to start with a really brief overview of the brain, which I think is really helpful. And as you can see here, the brain is really structured in different areas that fulfill different functions in controlling our activities. So you have, for example, the frontal lobe here on the left, which is really important for cognition and memory. Then you've got, for example, the temporal lobe at the bottom in blue, which is important for auditory areas as well as for long-term memory. Then you have the cerebellum at the bottom right, which is really very important for movement and coordination. Then you've got the occipital lobe on the left, which is important for vision. And at the top, you've got the parietal lobe that plays a really important role in integrating all the senses. If we consider the brain has about 100 billion nerve cells. So it's a vast amount of nerve cells, and they all consist of what we call a nucleus, and then we've got this long axon with the axon terminals. And you can see here the direction of how the messages travel from the body of the cell along the axon, then to connect to other cells. And the brain really is a vast network of lots and lots of nerve cells. And here, the sodium channels really play a really important part. And one of these sodium channels is the SCN2A sodium channel, and it's an ion channel. You can see here how that sits in the cell membrane of the nerve cell, and you can see here how the ions travel through the cell membrane to initiate what's called the action potential, and that's really the first kickoff to allow information to travel along the nerve cell, and then for information to pass from one nerve cell to another nerve cell. So it's really, really an important function for these ion channels to trigger and kick off the information transport from one cell to the next. And here on the right, you can see it's just a 3D model how these ion channels, here these sodium channels, sit in the cell membrane. You can see in the middle where there's a pore and the sodium ions traveling through. And I think what's really important to understand is that the sodium channel is a protein, and there's a recipe book for every protein in the body, and that recipe book is called the DNA. So the DNA is really the recipe book for any protein of the body, and that really contains all the information to build the structure of our body. And we've got a total of about 3 billion base pairs, which are these little sequences of information that make up the DNA. You can see here in the next slide is that a piece of the DNA that encodes the recipe for one particular protein, that's called a gene. So each DNA protein recipe is called a gene. And this really translates the information from the DNA into the building of proteins. And that's really important to understand, because if we have any spelling mistakes or what we also call mutations in the DNA, that leads to spelling mistakes in how we build a protein, and how, for example, if there's a spelling mistake in the SCN2A DNA or sequence, so the SCN2A gene, then we will get a spelling mistake and we might get a faulty SCN2A protein. And that's really the basics of understanding the genetics and terms of the mutations that might occur and how that relates to how the protein is actually formed. So when we then look at the sodium channel alpha 2 subunits, the SCN2A protein, so it's a voltage-gated sodium channel, and voltage means that it's triggered by currents that flow across the nerve cells, and it's widespread expressed in the central nervous system. It's got an important role in the initiation and conduction of action potentials, and it's expressed in the axon initial segments of the nerve cells and also in the nodes of Ranvier in early development. And it's really critical for normal early neuronal development. And here on the right, you can see how the ion channel is embedded in the cell membrane and how the ions travel through here. And so the structure is that of 4 domains that build the ion channel. So in the top right, you can see domain 1 to 4, and you can see below that each domain is formed in a similar way. So it has a voltage sensor that picks up the current across the nerve cell membrane in yellow, and then you've got the pore region, sections 5 and 6, and they all group together in domains 1, 2, 3, and 4 to build the ion channel with the pore in the middle. And we come back to this a little bit later. And so if you think about how the SCN2A gene is expressed in human tissue, so we know that the SCN2 gene is expressed mainly in the brain. So this is an illustration from how which genes, for example, are expressed across the entire body. And here you can see that this is mainly expressed in the areas of the brain. So, for example, in different parts of the brain, you've got different expressions of the SCN2A gene. That's really important, that it acts on different parts of the brain, and therefore it's really important for different functions in the brain. And here we talk about SCN2A related developmental disorders. So here you can see the illustration of the brain, and what we know, for example, that if you have mutations in SCN2A, you can, for example, have epilepsy. Yeah, and this is epilepsy you have if you have an uncontrolled excitatory signal in nerve cells that fire uncontrollably, and that might relate to an epileptic seizure. And this is, for example, work shown that if you have faulty SCN2A genes in, for example, your hippocampus, then this is this part in the brain that's highlighted here. You might then get seizures. However, also know that we have SCN2A that is expressed in the frontal cortex, so that's in the front of the brain. And we know that, for example, here what you can see is on the top left, you can see where it says wild type, you've got a sloping down, which is a wild type is a normal current that slopes down. Underneath, you've got a flat line where basically there's no current, there's a loss of function, and we know that loss of function SCN2A mutations are associated with autism spectrum disorder. Yeah, so we know that there's individuals who have a loss of function SCN2A mutation might present with autism. Lastly, you can see here that the cerebellum, which is at the bottom of the brain, and that's really important for movement control. So if you have mutations in SCN2A, SCN2A is also expressed in the cerebellum and might lead here to dysfunction or to the cerebellum not working properly, and therefore leading to, for example, episodes of ataxia or not having appropriate movement control. Now, this is a very nice paper collecting details from a really large cohort of individuals with SCN2A related disorders, and this was collected by Markus Wolf and Katrine Johansson in 2017, and it really was important to illustrate that we have 4 main groups of presentations within SCN2A related disorders. So we have the benign familial neonatal infantile seizures phenotypes. So Then we have the encephalopathy with early infantile epilepsy presenting at less 3 months of age. Then we have the encephalopathy with infantile or childhood epilepsy presenting after 3 months of age, which includes West syndrome, Lennox-Gastaut syndrome. And then we have intellectual disability or autism without epilepsy. And these are really the main types. And then Brunklaus and team looked at the different mutation classes in the SCN2A gene across their cohort. What they found is that the majority were missense mutations, where missense mutation, we know, leads to an altered protein function, but often it's not very clear how the child or the individual then present with that particular mutation, whereas if you have a truncating mutation, that often leads to a loss of protein function. And again, here, this is not always clear, the phenotype that this, also the clinic with which this child will present. And what we know is, and Dr. Brunklaus showed this, that if you have the early presentation of the benign familial neonatal infantile seizures, these tend to be exclusively missense mutation. In the later onset epilepsies, these tend to be also truncating but largely missense mutations. And in those patients presenting with intellectual disability and autism spectrum disorder, in 3/4 of presentations, these were truncating mutations, and the quarter were missense mutations. And so we know that for those presenting with SCN2A missense mutations, that almost 80% present with seizures, and the majority have seizure onset in the first weeks of life. And only about 20% of a clinical diagnosis of autism, autistic symptoms are reported. However, in the truncating mutations, we know that about 50% might have seizures, and the majority have seizure onset after the age of 2, and the majority have a clinical diagnosis of autism or autistic symptoms or developmental delay. And often doctors and geneticists and neurologists ask themselves whether a particular mutation is actually significant. And we know, for example, that in truncating mutations, that this leads to a loss of protein function, and often that is significant. However, in missense mutation, there's a number of other considerations. So in missense mutations, we really need to look a little bit in more detail at other information. So we want to know, for example, of this is a de novo, which means is this happened for the first time in a child, or is this actually inherited? Yeah. Then we want to know, does this particular mutation occur in the really important functional part of the protein? We want to know whether this is actually conserved in related species. If it's conserved, that highlights that this might be in a particular important area of the protein. Is there a particular difference in the amino acid composition? Do we have functional data? So this means, has this mutation actually been expressed in the laboratory to find out whether what the actual functional consequences of this mutations are? And we want to know whether this is actually present in the normal population databases. So this means if you have a missense mutation that is commonly found in normal individuals, is less likely to be significant as to if you have a mutation that's only found in individuals who actually present with disease. And how our genetic colleagues do this, there are particular tools they can use to classify whether particular mutations are significant or not significant. And what we know is that there's quite a number of sodium channel disorders and variants, mutations, where there's still some uncertainty whether these might be actually clinically leading to disease or not. So this is not trivial, and we know there's a number of variants who are clearly pathogenic. There's a number of variants or mutations that are clearly causing disease, but there's still a large number of variants or mutations where we're not entirely sure whether this actually causes disease. And it's really a team effort to come to terms and interpreting these results. So, and often the modeling that we have available is not really perfect to identify how relevant a particular mutation is. And what we have is an approach in our laboratory that we work very closely in a multidisciplinary team. So we have here child neurologists as well as clinical geneticists as well as genetic scientists. So the genetic scientists are the ones that perform the laboratory analyses, and then we meet together to discuss the genetic findings, and then the scientists will tell us, oh, we found this particular variant, we think this might be significant because of these properties of this particular mutation that we found, and then we can look at the phenotype or how the child presented, and we can then say, well, yes, this variant is in keeping with what we also find how the child presented. So it's really important to have this discussion between clinicians, this means doctors, either geneticists and child neurologists, as well as genetic scientists, to really discuss this in detail and to come to a conclusion as to whether a particular mutation might be relevant. And here, this is really looking at particular properties that we find in SCN2A mutations. So here we're looking at the early onset epileptic encephalopathy. So these are children presenting with SCN2A mutations in the first few weeks and months of life with really severe epilepsy. And these are often mutations that are not inherited from the parents and that happened newly in the child. And often, if they're early presenting, these tend to be gain of function mutations. So exactly here at the bottom, you have again this little cartoon of the ion channel. So for example, if the ions travel through the channel and the channel cannot close, then you have continuous flow of ions and you get too much firing off in the nerve cell. You get what's called a gain of function. And then if you have too much firing of the nerve cell, you get hyper excitability, too much. So that's too much activity, which expresses itself with an epileptic seizure. And the opposite is true if you have late onset epileptic encephalopathy and autism spectrum disorder. Here you have loss of function mutations, and these are in patients who present with later onset after 3 months of epilepsy, and in those with autism spectrum disorder. And for example, here the channel, for example, might not be able to open. Or the channel does not exist. And therefore, there's not enough firing in the nerve cell, and you get a loss of function. And there's a number of different voltage-gated sodium channels. We just spoke about SCN2A, but here what you can see from the top, so you've got SCN1A, then you've got SCN2A, 3A, and so forth. And I've highlighted here in darker gray those of the voltage-gated sodium channels that are expressed in the brain. So, for example, SCN1A is expressed in the brain, causes Dravet syndrome, which is another type of epilepsy. SCN3 is also expressed in the brain, causes also epilepsy. And SCN8A is expressed in the brain and causes also epilepsy. But SCN4A is, for example, expressed in the muscle, SCN5A in the heart, and causes arrhythmias, and then you've got SCN9, 10, and 11A, and they're expressed in the peripheral nervous system and cause, for example, inherited pain disorders. And if we think about the sodium channels, they're really quite closely related. And what I've here illustrated on the left is the evolution of the voltage-gated ion channels, and you can see here that they have a really common ancestry. And here you can see the ancestry is the same for potassium channels, and then calcium channels and sodium channels. The sodium channels are really, really closely related, and you can just see this is almost like the family tree of the voltage of the sodium channel family. What's illustrated here is how similar they are in their amino acid sequence. So the amino acids are the building blocks of any protein. And you look at the information, for example, between SCN1 and 2A, and there's only a 4% difference in the amino acid sequence between SCN1A and SCN2A. So it's showing that actually these proteins and sodium channels are really, really closely related. And just one slide just on SCN1A. So this is again a voltage gated sodium channel, and that is responsible for mainly for Dravet syndrome, which is again an early onset epileptic encephalopathy presenting in the first year of life, mainly with febrile seizures, and then other seizure types evolve. But also this has been associated with genetic epilepsy with febrile seizures, plus as well as with familial hemiplegic migraine, and also with other early onset epileptic encephalopathies. Then the SCN8A related disorders also presenting with epilepsy. And here we've got different phenotypes where we have benign familial infantile seizures, then the intermediate phenotypes, and then the severe presentation of developmental and epileptic encephalopathy. And what we know is that many of these mutations also cause gain of function, and that individuals may respond to sodium channel blocker use. This is very similar to SCN2A related disorders. And here, SCN3A related disorders, again, presenting with seizures very, very early on in life, but also with structural brain abnormalities. And again, this is also due often to gain of function in those SCN3A gene. And here just an overview of the different brain-expressed sodium channels. So you can see SCN1A, then SCN2A, SCN3, and SCN8 are highlighted here. The SCN2A and divided this into the early onset, which is less than 3 months, and later onset, which is after 3 months, presenting early onset with either the benign familial neonatal infantile seizures or the more severe early onset epileptic encephalopathies, often presenting in the first weeks of life with gain of function, and carbamazepine, sodium channel blockers, often are helpful. The later onset, after 3 months, presenting with West syndrome, with developmental and epileptic encephalopathy, presenting between 3 months and 6 months and 3 years, often with loss of function. And here, carbamazepine and sodium channel blockers are not really helpful. And what you can see here is that you have SCN1A, that's mainly a loss of function disease, whereas SCN3A and 8A, similar to the early forms of SCN2A, also often with gain of function, but SCN8A can also be associated with loss of function. I think it's really useful to present this just as a model of sodium channel disorders, and how these different disorders interact with each other, and the genes interact with each other. So what I've illustrated here is just a cartoon of nerve cells in the brain, and the effect that these nerve cells have. So as explained earlier, that all these nerve cells talk to each other, and there are different types of nerve cells in the brain. So in red, I've got illustrated the excitatory nerve cells. So these are the nerve cells that promote firing and pass on information. So you can see this here in red. And then you have the opposite are the inhibitory nerve cells that actually dampen down too much excitation. So they're expressed in yellow, and they then pass on information on other nerve cells that are here in gray, which are the corticospinal tract. So these are the nerve cells that reach out to the body. And if there's too much information passed on to these gray nerve cells, they lead to increased firing, which then might lead to epileptic seizures. And what you can see here is, for example, for SCN2, 3, and 8A, they're mainly expressed in these excitatory nerve cells, and if there's a gain of function, so which the upward arrow indicates here, there's a gain of function, too much function, then that excessive function is passed on to the gray nerve cell, leading to epilepsy. Whereas if you have, for example, in the yellow nerve cells, these nerve cells are mainly, the main goal is to control the other nerve cells. However, if you've got a loss of function in these yellow nerve cells, you're losing the control, and the red cells take over and and stimulate too much, and again you end up with gain of function. So with too much function. So, however, if you have these late onset seizures, and that's illustrated here on the right, and you have a loss of function in your excitatory neurons, often you end up with so you've got not enough output, and you can get a developmental phenotype of autism spectrum disorder. And I think what's important here is if we try to explain how these different genes actually interact, what's important to know is that these different genes and channels are expressed at different times in the evolution of the child and of the child's development. So what's expressed here is how much of these channels are actually built in the body at different times of the child's development. So on the left-hand side, you can see the expression of these genes and channels in prenatal form. So this is before birth. Then in the second column, it's from infant to childhood, then in the older child, and then in the adult. And here you can see in orange SCN2A expression, and you can see that this is really highly expressed even before birth. So SCN2A is one of these channels that's actually expressed very, very early on in life, which also means if there is something not right with that channel, this tends to present very early on in life. Yeah, and the expression of SCN2A remains high across the lifespan. What you can see here, for example, that for example, in purple, SCN3A is highly expressed in the antenatal form, so before birth, but then actually expression reduces with time. And what you can see, for example, for SCN1A, which is in green, this is not very much expressed as early on in life as a neonate, as before birth, but this then expression picks up in later childhood and adulthood, and similarly for SCN8A, which is not as much expressed early on, but the expression actually then rises later. And what we've done is we looked at this in a bit more detail and correlated here the gene expression with when seizures actually start in different disorders. And what we could see here is that to the far to the left, there's this SCN3A, and we noticed that here SCN. So what you can see as these circles, these display the gene expression strength. So how much of the gene is expressed, how early in life. And you can see here on the left-hand side, you've got the seizure onset from birth to about 6 years. And what we noticed for SCN3A on the left, so we have a seizure onset that is very, very early, and we've also got a gene expression that's very, very early. Yeah. So in SCN2A, what we find also that gene expression is very, very early, and also seizure presentation is very early. Whereas in SCN8A and SCN1A, we notice that the gene expression is later, and also the seizure presentation is later. So what we can show here is that really the gene expression is really tightly correlated with the seizure onset. Yeah, so if those genes that are expressed early and have a defect that then presents early, that means also that those individuals will present early with epileptic seizure. And here, this is just an illustration of how these genes are actually expressed together. And what we notice here is, and that's important on the left, you can see in the fetal brain, so this is in the unborn child, you notice that here SCN1A is expressed by itself, whereas SCN2, 3, and 8A all tend to be expressed to similar degrees, and these are all having similar function. So they're all act in similar ways. So just illustrating that SCN2, 3, and 8A in their functions appear to be quite closely related, which also seems to be reflected in their disease presentation. And here, what I think is important to illustrate how we can actually learn from the different positions of different mutations in across the SCN2A protein. So this takes you back to my initial slide, where you can see the 4 different domains. of the SCN2A protein. And here what you can see is so I've highlighted here different types of mutations that we find across the SCN2A protein. And what I've highlighted here are those mutations in green that are gain of function, so that lead to too much excitatory function and seizures, and are highlighted in red those that lead to loss of function. And here I've just illustrated a couple of mutations, for example, in red, where it leads to loss of function associated with autism spectrum disorder. And to the far right, there's green, there's a gain of function leading to developmental epileptic encephalopathy, so to severe epilepsy. And if we now compare the SCN2A mutations with, for example, SCN1A and SCN8A mutations, so you can see that these proteins are almost identical. And what we can now look at, different mutations that occur across different sodium channels. And we can learn lessons from this. For example, what I've highlighted here, so these mutations that occur at the same site in SCN2A might also occur in SCN1A. And what we learn is that those mutations that have been shown to lead to loss of function in SCN2A, if they occur at the same site in SCN1A, they also lead to loss of function in SCN1A. Yeah, and that's really important to know, because often we don't know what certain functions look like if we haven't tested them in the laboratory. And what we find here, for example, if you have a gain of function found in SCN2A at the same position SCN8A, that also leads to a gain of function. Yeah. So I think this is really, really important, because that gives us some idea, an estimation of what functional properties a particular mutation has if we don't have any functional data available. And we've done to a greater extent. So one of my research students has actually documented any mutation that has ever been functionally tested in any sodium channel gene, and plotted this here on this slide. And these are over 300 mutations. And this gives you a very good idea and roadmap of which areas of the protein are actually responsible for gain of function and which are responsible for loss of function. And this is really, really helpful to know, because if you come across a new mutation, you don't know for sure how to classify that mutation, this map might be quite useful to characterize that particular mutation, and to obtain some information about the functional consequences of that particular mutation. What I would like to highlight is a very, very interesting tool that has recently been developed by Henrike Heine and her team. And Henrike really looked at thousands and thousands of different individuals who presented with sodium channel and calcium channel mutations. And what she did is she plotted all of these and estimated the function of these mutations based on how these patients presented. And with this, she was able to create a map of the entire protein with an estimation of what this particular mutation, which functional consequences might be associated with that particular mutation. And that's really important. So this is now an online tool that is freely available. And with that tool, any clinician can use, for example, input a particular, for example, a particular SCN2A mutation to actually estimate what functional consequences this particular mutation might have, whether this might be, for example, a gain of function, or whether this might be a loss of function. And that might give the clinician an idea how to treat this particular individual. And I think this is just to illustrate that appropriate treatment appears to be important. So we know that in certain diseases, a certain particular drug might be indicated or might not be indicated, or a certain mutation or a certain particular drug might actually help to treat the epilepsy. So we know that in gain of function SCN2A mutations, often sodium channel blockers can be helpful. But we know, for example, that, so this is an example of SCN1A mutations leading to Dravet syndrome. But we know that in this disease, sodium channel blockers can actually be harmful. Yeah, because in this particular disease, it can actually make the epilepsy worse. And this is illustrated here by Iris de Lange and her group in the Netherlands. They looked at individuals who were treated with contraindicated medication in the first year of life, and they could find that if you didn't use the appropriate medication, or you used medication, in this case, sodium channel blockers for SCN1A related disease, where it was contraindicated, that those individuals in the long term did not do so well with regards to their cognitive development. Yeah. So similarly, we don't have any long-term figures yet for SCN2A related disorders, but if you have clearly an SCN2A related mutation, or an SCN2A mutation, for example, a gain of function mutation that responds to sodium channel blockers, we would hope that in the long term, the child would benefit from being on that particular medication. And of course, there's future opportunities for new treatments that are becoming available. And this really relates to all the sodium channel disorders, and these relate particularly to gene therapy approaches. And there's a number of different approaches that address this. And I think the idea here is that really you want to address the underlying problem of this of a particular mutation. For example, if you have an SCN2A gain of function, then you would like to have a particular therapy that can actually address that can actually address that particular gain of function and reduce the particular currents that are expressed excessively in these mutations. In other diseases, for example, in SCN1A, often these are loss of function diseases. So in that case, you try to achieve the opposite. You try to actually increase the expression of the gene, so you actually have got more expression. Yeah. So I think this is in the future, and now there's a lot of approaches in these different sodium channel disorders where we really try to target very precisely the underlying mechanism of this particular mutation, and then to treat that. And I think what's really important here is that we actually know the effect a particular mutation might actually have, and this is why these tools that I explained earlier might be of benefit, where you can actually estimate a particular function that a particular mutation might have from the experience of these accumulated knowledge of all these different previous studies or presentations. And we can use these tools to estimate function and thereby tailor our future treatment approaches. So this leads me to the end of my presentation, and I would like to conclude that voltage-gated sodium channel disorders really present according to the severity of the underlying gene defect, which is then relates in the clinical presentation, where there might be a mild or more severe presentations, that there's evidence that the symptoms and the different comorbidities that we find, that these are closely linked to the distribution of the gene defect across the brain, so that these genes affect multiple parts and aspects of the brain, therefore leading to a number of different difficulties and comorbidities, and not just epilepsy. And that we can see that some of these this heterogeneity, so the differences between different presentations often can be explained by the variant-specific gene expression, timing, and function. So we can see that different sodium channels are expressed at different time points and therefore also present at different times during development, and that the sodium channel variant characteristics can contribute to clinical decision making. So once you know whether a variant or a mutation will be at a gain or loss of function, that actually might help you to choose the correct treatment. Now, I would really like to thank my team in Glasgow, in particular my colleague Samir Zaberi, as well as Joe Simmons, but also my collaborators Dennis Lau in Cleveland, and Eduardo Perez, who's now in Chile, and all my students, and Stephanie Schorger in London. So this brings me to the end of my presentation, and I'd be happy to take questions.
There we go. And thank you very much for that presentation. So I'll open it for questions for everyone.
I hope I haven't overwhelmed you with details.
It was wonderful, thank you. Does anyone have any questions?
Don't worry if you think you might have a very simple question. Yeah, there's anything that you would like to ask, I'd be happy to answer. So I would say that if, so I think people will have, this is a lot of information that I've provided, and it might be worthwhile for delegates if they would like. I think this is available online as well, isn't it? So they can listen to this again, and because this is indeed a lot of information of years of experience condensed into a very, a very short presentation. But I'd be happy. So if there's, if there's follow-up questions, or some people would like to, for example, email Sally to say, oh, I've got a question about this, and then Sally, you can.
There's a couple here. There's Catherine says, I have a question about seizures in the womb. Would you typically see this in gain of function?
So yeah, so what we would expect is, so we know that SCN2A is expressed before birth. So we know that this is one of the earliest expressed sodium channels together with SCN3A. And if you have, and typically what we know is, if you have gain of function, those tend to be presenting the earliest in SCN2A. So I think, yes, if you have seizures in the womb, I think that would point towards that this might indeed be a gain of function, mutation.
And there's one from Tim as well. Does the current COVID crisis bring insights or opportunities for the type of research and development that you are doing?
The current COVID crisis, I think that's in terms of, so the one aspect is that COVID has actually facilitated this type of conference. So virtual working has actually, COVID has transformed virtual working, which has been very nice for us, because now we can actually reach out to far more people and have many more discussions like this virtually, and everybody's set up like this, which is really, really helpful. So I think that has helped communication in one sense. We've had also challenges in terms of research, in terms of COVID, because of course COVID took precedent over lots, for long periods of time, with regards to focus of research directed towards COVID-related research. So we've seen for a few months that our research efforts actually have been halted for a brief period of time, but that's now come to an end. So I think we're now catching up on our research efforts. So I would say that have been both give and take from the COVID experience we've had. But with regards to specifically research related to, I'm mainly involved in SCN1A research. You haven't seen any major impact of COVID onto that type of research.
Brilliant. Is there anyone else? I know the questions are taking a few seconds to pop through. He also says, but from the mechanisms used in the vaccine development?
Not for us directly, but you're right that, for example, the mRNA approaches and all that, we would hope that in the future this actually might give us a boost in terms of further knowledge of these techniques, which might actually be helpful. Yeah, that's correct.
Right, brilliant. Is there anyone else? Any last questions? I'll give them a few seconds, but yes, this session will be recorded and it will be shared with our private group, and we'll be able to share it as widely as possible, so the families can obviously take in the information as many times as they need to, because there was obviously a lot to take in, because it was a wonderful description of SCN2A and the other sodium channel conditions. So it was wonderful to have you on, Dr. Brunklaus. Is there anyone else before we? I don't think so.
Again, no, thank you so much for inviting me. It's been a pleasure to talk to you. And again, so I'd be very happy if there's questions in the future. I'd be very happy to answer those. If you would like to forward those to me, I'd be happy to get back to you. All right.
Fantastic. Thank you so much for your time.
That's great. Thank you.
Goodbye.
Bye.